Good morning, dear students. Today, one of my student by name Miss.Akanksha.K.IIICZBt from third year CZBT, she is presenting her seminar talk on in vitro construction of recombinant DNA molecules. So I request everybody kindly be with us with this particular seminar. If you have any doubts, we'll do that particular discussion and the queries will solve during the discussion class or do group discussion class. Now over to Akanksha.
Hello everyone, I'm Akanksha from third year CZBT. I'm here to present a seminar on the topic in vitro construction of recombinant DNA molecules. In this session, I'm going to cover the topics vectors, plasmids, the enzymes used in the recombinant DNA technology, the steps in recombinant DNA technology, isolation and extraction of foreign DNA, then the extraction of plasmid DNA from bacteria or any other cell, then restriction, digestion, and ligation of foreign DNA with the vector DNA, and finally the production of recombinant DNA molecule. Coming to vectors, these are very important prerequisites in recombinant DNA technology. So, what are vectors? Vectors are small, extra chromosomal, self replicating circular DNA molecules. For any molecule to call it a vector, it has to possess particular features. So, they are it has to be small and easy to isolate, it should be capable of replicating in the host cell, it should contain several marker genes which facilitate the screening of transformed cells. For example, the PBR322 plasmid vector has ampicillin and tetracycline sites. So these two antibiotic resistant sites are helpful in screening of the transformed cells, that is the cells which contain the recombinant DNA. And also the vector should have unique restriction cleavage sites. It should contain control elements like the promoters, terminators, etc. for the expression of the cloned DNA. So we have different types of vectors. So the most common are plasmids, bacteriophages, cosmids, expressons, bacterial artificial chromosomes, yeast artificial chromosomes. So I'm going to discuss only two of them as examples, plasmids and bacteriophages. In plasmids, these are again small self-replicating double-stranded circular DNA molecules. These are present in bacteria, yeast, and even mitochondria of some eukaryotic cells. So the special feature of these plasmids is they have genes for resistance to antibiotics, toxins, or toxic metals. Their size can vary from 1 kb to 200 kb. So the most common examples for plasmids are PBR322 and PUC19. So you can see the image of PPR322. So coming to the nomenclature of this particular plasmid, P denotes plasmid and BR denotes the name of two scientists, Bolivar and Rodrigues. And the number 322 is referred to differentiate this particular plasmid from other plasmids. The size of PBR322 is 4,360 base pairs. It has unique cloning sites like HIND3, BAM3, SAL1, PST1, etc. And it also has marker genes like ampicillin and tetracycline. So ampicillin and tetracycline, these are the two unique antibiotic resistant sites, and it has unique cloning sites like the HIND3, BAMH1, SAL1, PST1. So this is about PBR322. Now coming to PUC19. PUC19, the nomenclature goes like this. P denotes plasmid, and the letter U and C refer to the University of California. And this particular plasmid was constructed by J. Messing and J. Vera. And the number 19 distinguishes this particular plasmid from other plasmids. And this plasmid has ampicillin restriction, antibiotic resistance site, and it also has origin of replication initiation. It has unique cloning restriction cloning sites like BAM-H1, Hind3, and it also has eco-RI site. It has BAM-H1, etc. So this is about PUC19. Now coming to Now coming to bacteriophages. These are the viruses that infect bacteria. So the most commonly used cloning vehicles or vectors are phage lambda and phage M13. So this phage lambda usually infects E. coli T12 strain, the most common bacteria. And it also contains 48,500 base pairs. It has three regions, two essential regions and one non-essential region. The essential region contains the head and tail and the lytic functions, whereas the non-essential region is the replaceable region. So this is the genome organization of the lambda phage. Now coming to M13 phage. The M13 phage also infects E. coli cells. It has approximately 6,400 bases in it. To be precise, it has 6,407 bases, and it infects only F plus cells of the bacteria, and it doesn't infect the F cells. This particular phage is also double stranded, and it is also circular DNA. Now coming to some of the basic terms in rDNA technology. The most commonly used term is foreign DNA. What is foreign DNA? Foreign DNA is also called as passenger DNA. It is a fragment of DNA molecule which is enzymatically isolated and cloned from a particular species. So to explain it generally, it is a piece of DNA we are extracting from a particular bacteria or any other cell, and we are inserting it into the vector DNA. So in general, foreign DNA is nothing but the gene of our interest. Now coming to recombinant DNA. Recombinant DNA is the general name for a piece of DNA that has been created by combination of at least two strands. By saying combination of at least two strands, we refer to our gene of interest and the vector. So we are combining our gene of interest and the vector and forming a new DNA molecule, and we name it recombinant DNA or rDNA. Now coming to the technology behind this. So recombinant DNA technology refers to joining together of DNA molecules from two different species that are inserted into a host organism to produce new genetic combinations that are of value to science, medicine, agriculture, and industry. So in this technology, we are joining our gene of interest and the vector. We are inserting that into a host organism. We are producing new genetic combinations, and they are used in science, medicine, agriculture, and industry. And let me give you an example of how recombinant DNA works. I'm taking an example of producing synthetic human growth hormone. So basically, we are isolating the gene for human growth hormone from the human cell. At the same time, we are taking the vector, that is plasmid, from the bacterial cell. So we are then subjecting these two to restriction digestion. That is, we are joining our gene of interest, that is gene for human growth hormone, and the plasmid. So after joining, we are making them undergo ligation by using the DNA ligase. And then this particular recombinant DNA is inserted into a bacterial cell, and we're going to get the product, that is human growth hormone. Now coming to enzymes in recombinant DNA technology. In this technology, we're going to use a lot of enzymes to get the desired product. So we include nucleases. So nucleases are the first type of enzymes we're going to use here. In nucleases, we have three types of enzymes: restriction endonucleases, restriction exonucleases, and ribonuclease H. So restriction endonucleases and exonucleases are used to cut DNA into fragments of a size suitable for cloning. So we are going to use these two to cut the DNA to form uh, to get the gene of interest. And then ribonuclease H is used to cleave RNA backbone phosphodiester bonds to leave the 3' OH group and 5' phosphate group. The second type of enzymes we are using is DNA modifiers. All these listed uh, enzymes are used to modify DNA in the desired way. So, DNA polymerase is used to synthesize cDNA, that is the complementary DNA. Reverse transcriptase is used to fit the RNA genome into the host DNA genome. Basically, this is present in RNA type viruses to undergo reverse transcription. Now, coming to alkaline phosphatase, this particular enzyme removes 5' phosphate group and prevents the ligation of the vector. And then polynucleotide kinase. It is used to convert the 5' OH into 5' phosphate. Terminal nucleotidyl transferase adds deoxynucleotides to 3' OH of DNA molecules. Then methyl transferase adds methyl group to adenine or cytosine bases within the recognition sequence to protect from endonuclease. The type 3 enzymes are the DNA ligases. These are used to ligate our gene of interest and the vector. Coming to the applications of rDNA technology, as I already said, we are taking the gene of interest and then inserting it into a plasmid from bacteria, then making the recombinant plasmid or the recombinant DNA, and then we're inserting it into a host cell for producing the desired product. And then the desired product can be an amplified protein or an amplified gene. It is used in scientific research, medicine and pharmacological industry, pest or insecticide resistant plants, and this bacteria is also used to clean oil or toxic spills. If it is an amplified gene, it is again used in scientific research, medicine and pharmacological industry, high temperature forming snow, and food industry. These are some of the examples of applications of rDNA technology. Coming to the steps in recombinant DNA technology. So first thing, to get the gene of interest, we need to isolate that gene of interest from a particular species. Then we're going to subject that gene of interest to restriction enzyme digestion, and then ligation of DNA molecules and insertion of recombinant DNA into host. So there are basically four important steps in rDNA technology. Now coming to the first step, that is isolation and preparation of foreign DNA. So to extract DNA from a bacterial cell, the cells are first treated with an enzyme called lysozyme in order to weaken the bacterial cell wall. So the bacterial cell wall is thick and tough. So to rupture that, we are going to treat it with another anionic detergent such as sodium dodecyl sulfate. Then we come into eukaryotic cells. Plant cells have a rigid cell wall. So to treat that cell wall, so to rupture that cell wall, we are treating with cellulase or any other abrasive as such to rupture the cell wall. And then the animal cells do not possess cell wall, they only possess cell membrane, that is plasma membrane. So to isolate DNA, we are again subjecting it to sodium dodecyl sulfate, and we're going to rupture the cell membrane and solubilize it. Now, the DNA in the lysate can be extracted by suitable extraction procedure. The procedure goes like this. The first step is the extraction of bacterial DNA. So the bacterial cells are added into an extraction medium containing saline EDTA. So here the function of saline EDTA is to uh, in saline DTA, the salinity is to prevent strand separation. We want the double-stranded plasmid to be double-stranded DNA to be like that. We do not want to separate it. So to prevent the strand separation, we are providing a counter ion shield on the double helix of the DNA. And EDTA will ensure the chelation of metal ions. So the metal ions will form a ring around the DNA so that it doesn't undergo denaturation. And we're also subjecting it to 10% STS in the next step. We are incubating it at 60 degrees Celsius for 10 minutes, and then we are cooling it on ice. The next step is addition of sodium chlorate solution. We are adding sodium chlorate solution for protein denaturation. We are going to destroy all the protein present in the cell to extract DNA. Then we add chloroform and isoamyl alcohol. So isoamyl alcohol is an anti-foaming agent. It doesn't form foam while extracting DNA. And chloroform helps in the extraction of DNA easily. Then ethanol and isopropyl alcohol. The ethanol and isopropyl alcohol help in purification of DNA. Then the next step is first ethanol precipitation. So we are going to subject the DNA, the lysate which we got in the previous step to centrifugation. The centrifugation is done at 10,000 rotations per minute. Here we are removing the upper aqueous layer and we're keeping it in a beaker, and we are going to overlayer it with 80% ethanol. Now these two layers are shaken with the help of a glass rod, and the DNA fibers are wound out on the rod. And we can see them like fibers. Now we're going to remove the solvent. Finally, fibrous DNA is blotted using filter paper. Now we're going to subject this to RNase treatment and second ethanol precipitation. This is the third step. In this step, we're going to add saline sodium solution consisting of sodium citrate and incubate it for 30 minutes at 37 degrees Celsius. Now, the function of saline sodium solution is the dissolution of DNA. We're going to dissolve the DNA now. And sodium citrate will check the activity of DNases so that all the DNases will stop performing and the DNA will be dissolved now. Then we are incubating it with RNase. The function of RNase is to ensure degradation of contaminated DNA. We want only DNA. We do not want any RNA in this extraction. So we're going to destroy all the RNA that is present in the lysate. And then we're going to add sodium chlorate solution. The sodium chlorate solution will stop the reaction of RNase. And then chloroform and isoamyl alcohol are added. Now we're going to remove all the solvent by 80% by adding 80% ethanol, and DNA is pulled out by glass rod. We can actually see it like fibers. This is then purified and stored at minus 40 degrees Celsius. Now the last step uh, uh, is the isopropanol precipitation. We are going to add isopropyl alcohol to precipitate the residual DNA. And then this DNA that we got from the above step is redissolved in sodium saline citrate, and it is stored at minus 40 degrees Celsius. Now we're going to come to assay for nucleic acids. So now whatever DNA we got from the previous step, we're going to subject it to spectral analysis. Here it is UV absorption spectroscopy. So here we're going to consider that 1 gram per ml of DNA gives absorbance of 2 at 260 nanometers taken in 1 centimeter cuvette. So this is the standard we have fixed. Then the ratio of E260 to E280 should be between 1.8 to 1.9 for pure DNA. And a value less than 1.5 says that there is protein contamination. Which to ensure this, it should be 1.8 to 1.9. Now, till now, we extracted the gene of interest. That is, we isolated the gene of interest. Now we're going to extract the plasmid DNA, into which we're going to introduce our gene of interest. So for this, we are taking the culture of cells in a liquid medium. We're going to harvest and the cell extract is prepared. Now the cell extract is deproteinized and RNA is removed. So we're going to remove the protein and RNA, and the DNA is concentrated by ethanol precipitation, as I already said. Now that we've got DNA now that we've got the bacterial cell, it has both plasmid DNA and the bacterial chromosomal DNA. Now our duty is to remove only the plasmid DNA for our experiment. So we can separate the plasmid DNA from the bacterial chromosomal DNA in two ways. One, separation on the basis of size, and the other is by separation on the basis of conformation. So on the basis of size, first we're going to perform cell lysis. Then the DNA fragments are removed with cell debris by centrifugation. Now the cells are treated with EDTA and lysozyme and then Triton X100. So the Triton X100 is mainly responsible for the cell lysis. After centrifugation, now the cleared lysate contains almost entirely of the plasmid DNA, and that is what we want. Now, the second is the separation on the basis of conformation by alkaline denaturation. So we're going to use alkali here, that is sodium hydroxide, and the pH should be maintained between 12 to 12.5. This sodium hydroxide is added to the cell extract, and the pH should be between 12 to 12.5. There is a reason for this. The reason is that supercoiled DNA, that is our plasmids, can be easily removed from the non-supercoiled DNA, that is the bacterial chromosomal DNA. So the in a pH between 12 to 12.5, we can denature the non-supercoiled DNA, that is the bacterial chromosomal DNA. So this is done to extract our interested plasmid. So if acid is added, then the denatured bacterial DNA plasmids will re-aggregate into a mass. Now this insoluble network can be pelleted by centrifugation, leaving plasmid DNA in the supernatant, and RNA and protein are removed by centrifugation steps. Now that we are done with extraction of our gene of interest from the bacteria and the plasmid, we're going to subject this to restriction, digestion, and ligation of foreign DNA with the vector DNA. This covers the third and fourth step of the recombinant DNA technology. In this step, we're going to use restriction enzymes to cut the gene of interest and the DNA ligase. So most commonly used restriction enzymes are type 2. We are not going to use type 1 and type 3 in this. We are using type 2 restriction enzymes, which creates staggered or blunt ends. So both the donor DNA, that is our gene of interest, and vector DNA, that is plasmids, are digested with the use of same restriction enzyme that produces sticky ends. And now these are mixed in a test tube to allow the sticky ends of vector and donor DNA to bind to each other to form recombinant DNA molecules. So we want both of them to join. So for that, we're going to use DNA ligase. So basically, a plasmid vector carries a single eco-R1 restriction site. So digestion with the restriction enzyme eco-R1 converts the circular DNA into a linear molecule with sticky ends. Now, the donor DNA from any other source, that is our gene of interest, is also treated with the EcoR1 enzyme to produce a population of fragments carrying the same sticky ends. Now, when the two populations are mixed, the DNA fragments from the two sources can unite, they join, and because they have double helices and they form sticky ends. There are many opened up vector molecules and different fragments are formed. So we can therefore we can see diverse array of vectors carrying different donor inserts in the mixture. So now at this stage, although stichians have united to generate a population of chimeric molecules, the sugar phosphate bonds are still not complete at two positions at each junction. So now the backbones are sealed by addition of the enzyme DNA ligase, which creates phosphodiester bonds at the junctions. Certain ligases are even capable of joining DNA fragments with blunt ends. In this procedure, I've only told about the ligation of sticky ends, but certain ligases can also join the DNA fragments with blunt cut ends. Now, finally, coming to the production of recombinant DNA molecule in large scale, that is in the industry. So first we're going to select the donor DNA and the vector DNA and purify a gene sequence to be transferred. Now cut the larger donor DNA and the vector DNA with same restriction enzyme to get same kind of sticky ends. Now the modification of cut ends is done. Then we're going to mix both the donor DNA and the vector DNA and add T4 DNA ligase to join the sticky ends of gene of interest and the plasmid. Now we are amplifying the recombinant DNA in E. coli cells. The screen's clones are transferred to fresh culture plates and grown into large populations. Now we're going to screen clones containing the recombinant DNA molecules. We're going to extract recombinant DNA from cells and purify it, and then this recombinant DNA is used for various genetic engineering applications. So in general, so we have the vector DNA and the donor DNA molecule. It has the target sequence. So now we are going to cut both the gene, both vector and the donor with the same kind of restriction enzyme. Now we're going to modify the cut ends by adding linkers for better joining, and then we're going to mix it, and we're going to add T4 DNA ligase, and the recombinant DNA molecules are formed. They're introduced into E. coli, and we're going to culture those cells on agar plates, and then finally selection of rDNA, and then the clones containing recombinant DNA, and we're going to grow in fermenter to recover foreign proteins. So these are the references I've used. So thank you.
Can we go to the first slide, Akanksha?
Yeah, ma'am, sure. Yeah, ma'am.
Okay, good job, Miss Akanksha. Done really well.
Thank you, ma'am.
Okay. You have done a good job, good presentation.
Thank you, ma'am.
Okay, you're welcome. Further, you will be notified your seminar marks very shortly based on your presentation, voice modulation, and content. But you have really done a wonderful job.
Thank you, ma'am.
Thank you. Okay, next you get ready for your paper 6, that is immunology and animal biotechnology in another session. Okay, and whatever the updates we'll discuss in the GD, that is group discussion class, or maybe in the interaction class. Okay, and thank you for your presentation.
Thank you, ma'am. Thanks for giving this opportunity to present.
You're welcome. You deserve. Thank you.